Okay, good morning, everybody. I am going to talk to you about using data science, and I can't see, okay, using data science and molecular modeling to predict pharmacotherapies pretty much to fight drug addiction. And trying to advance my slide and it's not advancing.
Just click, Dr. Keith. Maybe that will do.
Yeah, it's clicking, it's not going. Okay, let me, oh gosh. And I can't see the tabs to let me advance.
Do you have two monitors or one monitor?
I have one monitor.
Okay.
And I'm trying to leave full screen because if I leave full screen, I think it's better.
Yeah, then go with that, I guess.
Let me stop my share and I'll start sharing it again. Okay. See if that helps. No, it's not doing it. Yeah. Oh, there we go. Okay. Okay. And so, apologize for that, but we have several unmet medical needs because of chemical imbalances in the brain. And I've shown here some neurotransmitters, dopamine, serotonin, and norepinephrine. And when there are some imbalances, they cause disorders such as depression and the other ones I have here. However, we're going to focus mostly on drug addiction because this is a huge problem. Now it's doing it again. It is not advancing.
Do you want me to run it from my side?
Yeah.
Okay, so I'm sharing my screen then.
Okay, I have to stop sharing.
Can you all see my screen?
Yes, we can. Yeah, my directional keys are not working. I see something in the chat. They're just not working. Okay.
So are you in this slide?
Move on to the next one. Okay, so drug addiction is not considered to be a it's considered to be a neuropsychiatric disorder. It's not a moral problem. And we know that it costs a lot of money to taxpayers just for cocaine abuse, but the human suffering we cannot really put a price on. And right now we don't have any pharmacotherapies for cocaine addiction. We only have behavioral therapies. Next slide, please. And so we're going to tackle this by looking at what is happening in the brain in the presence of cocaine. So usually your neuron will release dopamine into the gap between the neurons. And this is a normal process of it binding to the receptor, getting the physiological response, and then having reuptake back into the original neuron. It's like you going to the store, picking up some things, going back home, okay? And then if cocaine is there, it blocks the door for you to go back home. And so what happens is this has a lot of dopamine in the synapse, and this is causing heightened euphoria. And people become addicted to that. Next slide, please. And the traditional way of finding a drug to manage disorders is taking 15 years to do all of the studies in the animal, outside the animal, and in people. And it costs about $1 billion, take or give a couple of $100,000. Next slide, please. And so our approach is to accelerate this process by using data science. And some people use artificial intelligence, but we know that we are looking at the portion that involves data science for this. Next slide, please. And you can see here that artificial intelligence has impacted drug discovery in many ways. I am going to focus on what's in the boxes here. So I'm going to look at it with drug design and also look at it with drug repurposing. We also use drug screening, but I'm not going to talk about that, but they all go hand in hand. They're complementary to one another. Next slide, please. And so what we do is look at publicly available data, and we correlate gene expression based upon how the cells react when there is when they're healthy and when they're not healthy. And so we look at what genes are upregulated or what are expressed more and which ones are downregulated. And we compare it to the healthy cell and the cocaine treated cells. And this is publicly available data, and we have a lot of algorithms that we can use. And we have to just choose them wisely and choose the data wisely that we're looking at. Next slide, please. And again, I'll point you back to this slide because what we're doing is looking at rats. So we're starting small, just 10 rats, and looking at what happens to the rat when it is treated with cocaine and comparing it to the neurons of the rat when it's not treated with cocaine. So that's the control. And we look at the difference between the two. So even though the brain is very complex, there are a lot of brain regions. We are focusing on the nucleus accumbens and the prefrontal cortex, because the nucleus accumbens is the site of your dopamine, dopaminergic cells or your dopamine cells. And we're going to compare it with the prefrontal cortex because we have to look at the differences in gene expression between those two areas because they are most implicated in drug addiction. Next slide, please. And so in our set that we chose, we have 5 brain areas that were studied, and we had 10 rats altogether. And you can see we have cocaine treated and saline treated, which is nothing but salt. So that's why it's the control. Next slide, please. And we wanted to look at only the nucleus accumbens, but understand that the reason why there are those other regions is because dopamine reward system impacts other parts of the brain. And so we'll go back and look at that later, but we want to focus solely on nucleus accumbens and the prefrontal cortex, which is near the caudate pituitary, which is in the front, but it's not on this slide. Next slide, please. And when we look at gene expression, we have to compare it based upon the color that we see and based upon the intensity of that color. So when you have no color at all, that means that there was no effect at all in that brain area of the rat for that sample that was looked at. So we had 8,523 experiments. So those are the data points that we are working with. And we are focusing only on the nucleus accumbens and the prefrontal cortex, as I said, but this microarray does show the 5 brain areas, and the intensity shows how much upregulation there is and the significance of that regulation. We'll look at that as well. Next slide, please. And when you look at the microarray experiments and look at what's upregulated, what's downregulated, we find that there are 250 genes actually that are most important. And the top 16 is where we're going to focus ours when it comes to correlating with possible therapy. And the Volcano plot here shows what genes are upregulated, what's downregulated, and it's showing the intensity and also the significance. And so this is often used to help us see what genes we need to focus on. Next slide, please. And the top 16 genes that we have, you'll notice there that the dopamine receptor is important. It's showing up twice, okay? And there are some other ones that show up twice. However, we know that others have targeted the dopamine receptor in finding a pharmacotherapy. So that's important for us to as a starting point. And there's some other important genes there, but I won't discuss those. Next slide, please. And so now that we have that piece of information, we now have to look to another way of applying algorithms to data when it comes to the FDA-approved drugs. Next slide, please.
Dr. Keith, you have 1 minute left.
Okay. And so this is the other website that we use, and we can choose based upon the genes. If you see there, it has targets. And so we have different genes here that we can prove, that we can choose. And so we see the DRD1, but we also see the solute carrier gene. Next slide, please. And that was a little bit more important. And when we look at some of the studies that have been done with diphenylpyraline as a piperidine, it is less likely to be addictive, but it also can replace cocaine. So we said, well, let's look at that and look at the gene targets. And of course, it's coming up with the solute carrier, which is in the top 250, but not in the top 16. And because it's a anti-allergen, it attacks histamine as well. Next slide, please. And then we used AI and looked at the correlation and why it would bind to the dopamine transporter. Next slide, please. And wanted to acknowledge the team who's works with us on this, on CADS. And the Center Projects here was written up in the journal article in the Winston-Salem Journal, March 26th. And I'm running out of time, so next slide, please. Just want to acknowledge also others from the team and where our funding is from. And next slide. And want to thank you for your time.